So, all right. Well, John, they're still coming in. We want to give it just about another 5 seconds. Let's give it 10, 15 more seconds, Doctor, if you don't mind. And because they need to hear what you have to say. I mean, with what we saw yesterday, it's just imperative that they don't miss out. And it's just not the same when they listen to the recording as being live. So.
Yeah, Keith, it's John. I'd like to begin by thanking all the people on the call today for taking the time to be with us. I'd like to especially thank Keith and John for putting this series of Zoom calls together. It's been a great platform for us to highlight important aspects of Immunotech's unique story. And on the last Zoom call, I closed my portion with the following thought. If you look at the events that led to the discovery of Immunocal, the formation of Immunotech, the commercialization of the product, it happened because highly talented people worked together as a team towards a common goal. Now, this is a great lesson because nobody has all the answers. So surround yourself with people who share values, passion, and vision as we did, and great things can happen. Now, ladies and gentlemen, this could not be more on point when discussing the contributions made by our next speaker. On Friday, January 23rd, 2009, at 1:27 p.m., I received the following email from him. Thank you for the interesting information on Immunocal. I would definitely be interested in obtaining some of this initially to try some in vitro experiments in primary neural cultures to see what effects on glutathione status and neuroprotection we observe in cell culture models of neural apoptosis and oxidative damage. Please let me know how to go about obtaining Immunocal for these types of experiments. So little did I know at the time when I was reading that email, the critical role this scientist would play in helping us develop our research platform on Immunocal and brain health. Besides publishing a wide array of papers, he has contributed to 2 of our patent applications, one in traumatic brain injury, and the second one based on our Reelin data, which you'll hear about later. He's also presented Immunocal data at a number of conferences, including the 2018 Alzheimer's Association International Conference, the 2018 Annual Traumatic Brain Injury Conference in Molecular Biology, as well as the Keystone Conference, where he presented our ALS data. Dr. Lindsman is a professor of biological sciences and associate executive director of the Nobel Institute for Healthy Aging at the University of Denver. Dr. Lindsman teaches courses in neuropharmacology and cell signaling and performs research focused on neurodegenerative disorders and neurotrauma. He received his BS in biology and chemistry from the University of Michigan in 1987. He then worked 8 years as a research technician at the Upjohn Company in Kalamazoo, Michigan. He went back to the University of Michigan in 1996 and completed his PhD in pharmacology. He did his postdoctoral training from 2000 to 2005 at the Denver VA Medical Center and received his first independent VA grant in 2006. He held a joint appointment with the VA and the Denver University from 2006 to 2015 and has been full-time on the faculty at Denver University since 2015. His professional affiliations include the Society for Neuroscience and American Society for Biochemistry and Molecular Biology. Dr. Lindsman has published 76 times to date, with many more to come based on his scientific curiosity. Dr. Lindsman is a highly valued member of our Scientific Advisory Board. Ladies and gentlemen, it gives me great pleasure to welcome Dr. Dan Lindsman to our Zoom call today.
Hi, John. Well, thank you very much for that really kind introduction, and I want to thank John for inviting me today, and thank you all for attending. I've been working for quite a few years on oxidative stress and neuronal apoptosis and how those pathways are involved in neurodegeneration and neurotrauma. And so Immunocal was an obvious fit for this research. So what I'd like to do today is to take you through some of our published works and then also through some data that is not yet published in an Alzheimer's model, so that you can get an idea of the kind of the vast experiences that we've had studying Immunocal. So I'm going to share my computer screen here. And hopefully you can all see that. Yes?
Yes, we can, doctor.
Great, great. So this is one of the first studies that we started years ago. I can't believe that 11 years has gone by, really. But we initially were looking at in vitro models or cell culture models to see how Immunocal, number one, could it protect nerve cells or neurons from different stresses, particularly oxidative stress? And if so, what was the mechanism underlying that neuroprotection? And so I think with this, I'm going to start actually with the last figure in the paper so that I can give you an idea of what the summary really is. And so my laboratory focuses a lot on the mitochondria and the role of mitochondrial oxidative stress in neuronal cell death, in neurodegenerative disorders, and neurotrauma. Many different types of insults or stressors can impinge on mitochondrial function, which are really your powerhouses of the cell. And this damage that's done to the mitochondria not only decreases energy supply, but it also causes oxidative damage, which can lead to cell death. And what we showed in this paper, sort of in a nutshell, is that Immunocal, by providing a precursor, cysteine, for glutathione synthesis, actually suppressed neurotoxicity or provided neuroprotection from a large array of stressors that act primarily at the level of the mitochondria. So this includes things like free radicals like nitric oxide and peroxide, heavy metal toxicity like that induced by copper, aluminum toxicity, which aluminum is a very well-known neurotoxicant, as well as toxicity caused by, in this case, amyloid beta peptide. Some of you might be familiar with that. This is the peptide that makes up amyloid plaques in Alzheimer's disease. What I want to talk about briefly, just to show you a little bit of the data from this study, is this here. It's called a BCL2 inhibitor. BCL2 is a key pro-survival protein that protects the mitochondria from oxidative stress and really limits what we call neuronal apoptosis, or programmed cell death of neurons. Now, I can show you some pictures of what happens when we treat neuronal cultures with this BCL2 inhibitor. So in the left panel here, these are neurons actually taken from rat brain. These are stained for the nucleus, so this is the DNA, the chromatin of the cell. In the middle panels are the neuronal processes, so these are the networks of connections between these neurons in the dish. And the right-hand panel is the merged image, so the blue are the nuclei and the green are the neuronal processes. And what you can see in this first panel, these are healthy control neurons, and you can see they form this vast interconnected network, and these cells are very healthy. What happens when we add a BCL2 inhibitor, I think it's quite dramatic, right? The cells essentially get destroyed. The nuclei become very small and punctate because they're undergoing apoptosis. And the neuronal processes essentially are completely disassembled, right? So now there are no connections between neurons. But this next finding is really spectacular in my mind. I've been doing these kind of studies for about 20 years, and I've never seen something that protected neurons as well as Immunocal. So here we treated the cells with Immunocal and the BCL2 inhibitor, and you can see all of the neuronal processes are maintained and the nuclei are still large and healthy. And so as we would predict, what happens if we measure glutathione in these neurons, we see that in response to the BCL2 inhibitor, we get a dramatic reduction in cellular. Okay, we're back. Okay, I apologize for that. I don't know what happened there. So what I was saying here is that we were trying to look at the mechanism of how Immunocal protected these neurons so well from this BCL2 inhibitor. And what we found was that, as predicted, this compound dramatically depletes cellular glutathione and induces tremendous amounts of cell death. However, when we treat with Immunocal, we preserve or protect that cellular glutathione, and that, of course, then reduces the cell death dramatically and protects the neurons. But how do we mechanistically show that those two events, the preservation of glutathione and the neuroprotection, are connected, right, mechanistically? Well, in a cell culture system, you can do that because we can put in a compound here, BSO or buthionine sulfoxamine, that actually inhibits the synthesis of glutathione from cysteine. And when we put in that compound, you can see it blocks the ability of Immunocal to preserve glutathione, and you lose the neuroprotective effect completely. And so this was really the first study in vitro that we were able to very convincingly show that the ability of Immunocal to protect neurons from different types of oxidative stress-inducing agents is absolutely dependent on its ability to stimulate or preserve glutathione synthesis. So we went on from these in vitro studies to do a number of different studies in mouse models of various either neurodegenerative disorders or neurotrauma effects. So the first study that we did was in a mouse model of amyotrophic lateral sclerosis, or ALS. This is a transgenic mouse that expresses a human mutant transgene of the SOD1 gene. This mutation is found in a small percentage of familial ALS patients. So what we did is we gave these mice Immunocal either as a direct, what we call oral gavage, where we basically put a tube in the mouse's mouth and deposit the Immunocal directly into the stomach, or we gave the Immunocal in their water so that they could just drink it on a daily basis. And either way, we got the same effect in this mouse model. What we saw was about a 10 to 12 day delay in the average onset of the disease in these animals. And we measure this onset through changes in motor function, as ALS is really a neuromuscular disease that causes deficiencies in motor function. Now, a 10 to 12 day delay in disease onset may not sound very significant, but you have to understand that in a mouse, 10 to 12 days is really roughly equivalent to 18 to 24 months in a human. So this is actually a very large effect in this mouse model. We also looked at motor function in these animals, and one of the ways that we test motor function is by something called a grip strength test. Basically, you take the mouse, you put it on a wire grid, and you invert the grid and see how long the animal can hold on with its hind legs, its hind paws. Now, mice are tremendously adept at this. This top line are the wild type or non-transgenic healthy mice. We stopped this assay at 30 seconds, but they could hold on for hours, literally. They climb around on it upside down. They have no issues. This line here, though, are the mutant transgenic mice that have ALS that were untreated. And you can see by the time they're about 105 days old, essentially these animals have no strength in their hind limbs, and they're not able to hold on to the wire grid. And this is really due to a loss of innervation of the motor neurons into the skeletal muscle in the legs. This is part of the pathology of ALS. What you can see is that the animals that we gave Immunocal to, they showed a really nice preservation of motor function here. So this is skeletal muscle strength in their hind limbs. And this is a very big difference. You can see that they sustained really this increased muscle function out to nearly 120 days old, which is normally about the end stage where these animals start to succumb to the disease. We also looked at glutathione in the spinal cord. So this is actually looking in the lumbar spinal cord, which is your sort of your lower back, where the motor neurons project their axons over 1 meter in length to innervate your large leg muscles. And what we were looking at here is we used a technique called HPLC, or high performance liquid chromatography, to measure both the reduced and oxidized forms of glutathione. What we found was that in the spinal cord of these animals, compared to the healthy non-transgenic mice, ALS mice show a marked reduction in the reduced form of glutathione and a marked increase in the oxidized form. This leads to a decrease in the ratio of reduced to oxidized glutathione, and this decrease is really reflective of tremendous oxidative stress in the spinal cord of these mice. But what's quite remarkable is the animals that we treated with Immunocal, you can see they showed a preservation of the reduced glutathione and essentially no increase in the oxidized glutathione. And so this led to really a clear protection of the reduced to oxidized glutathione ratio in the spinal cord, indicating that Immunocal treatment actually suppressed oxidative stress in the lumbar spinal cord of these ALS animals. So this was a pretty fascinating finding. The last thing that I want to show you from this paper was kind of an interesting study that we did. So if any of you are familiar with ALS, you'll know that it's really a severe neuromuscular disease. There are very few drugs, only 2 now approved for ALS, and they really don't have very large effects in terms of therapeutic effects. The one drug that was approved all the way back in 1995 is riluzole. And riluzole has what we call an anti-glutamatergic action. It basically decreases calcium-mediated toxicity in the motor neurons. So quite a few patients that have ALS, probably about 50% of all ALS patients, are on riluzole. And so one of the things that we wanted to make sure was that if we gave these patients Immunocal, that we would not in any way interfere with the actions of riluzole. Even though they're not tremendously effective, there are some positive effects of riluzole. And so what we did is we either treated our ALS mice with riluzole alone or with Immunocal and riluzole. And what we found here is that the combination of the two, we still see this delay in disease onset. You can actually see that you do not see this with riluzole. It doesn't have this effect, so this is actually the Immunocal effect, delaying disease onset. But more importantly, riluzole does extend survival of these animals, and the addition of Immunocal had no adverse effect on the survival promoting action of riluzole, indicating that patients that are on riluzole and receiving some benefit from this drug would not have an adverse effect from taking Immunocal on top of that. And that's a very important finding. So that was our ALS studies, and through conversations with John Molson and that, we decided to focus in on neurotrauma. As you are probably aware, concussion and traumatic brain injury is a tremendous unmet medical need in the health community. There's approximately 1.7 million concussions every year in the United States, and there are no drugs approved for the treatment of concussion. Now, we were particularly interested in high-risk populations that are in either occupations or that have a high risk of concussion. So this would be athletes in contact sports, military personnel, as well as the elderly, because the elderly, unfortunately, do have a lot of concussions due to their propensity to fall. And so we actually did a study using a mouse model of concussion or traumatic brain injury, in which we use a model called controlled cortical impact. And basically what this model is that we, under anesthetic, we do a traumatic brain injury to the skull of the animal that would be comparable to somebody having a moderate concussion. So this might be an athlete playing football and having a moderate concussion, akin to maybe just a slight loss of consciousness, maybe for a couple seconds. And what we did is, because we were interested in seeing if Immunocal could increase resilience against the negative effects of concussion, we actually pre-treated these mice with Immunocal in their drinking water for about 4 weeks prior to giving them a concussion. And then we looked at a whole host of behavioral assays, histopathology, as well as biochemistry to analyze how Immunocal affected the response of these animals to concussion. And so I'm not going to take you through all this data. There's a lot of data in this paper, but I do want to highlight a few things. The first thing here, you're going to see two graphs. One is called writing reflex time, and the other one is apnea time. So apnea refers to the inability of the mouse to breathe. So once under anesthesia, when we do the concussion on these animals, for about 30 seconds, they stop breathing. Okay, and this is common in these types of paradigms that we use in mice. Writing reflex time refers to how long the animal takes to wake up from anesthetic after the concussion. The sham animals here are the controls. Essentially, they have all the same surgical interventions that we do to the animals that get the traumatic brain injury, except they don't get hit, right? So they are a control, but they don't ever suffer a concussion. What you can see is that the sham animals take a couple minutes to wake up from anesthetic, and of course they don't have any apnea at all. The animals that were untreated that received concussion, you can see they take quite a bit longer, more like 6 to 7 minutes to wake up from anesthetic after receiving the concussion. And they undergo about 30 seconds of apnea. The important thing here is that the animals that were treated for 30 days prior to the concussion procedure with Immunocal, they don't have any difference in either their writing reflex time or apnea time from the animals that did not receive Immunocal but received concussion. So why would this be important? This is important to demonstrate that both the untreated and treated mice had the same magnitude of concussion, okay, induced. There was no difference. We didn't hit the Immunocal animals with a lighter magnitude of concussion, for example. So this is important sort of control data to make sure that it's a fair test of the effects of Immunocal. So then we did several behavioral assays. One of those was something that we call a beam walk. And basically this is the ability of mice to walk on an increasingly narrower and narrower beam. Okay, mice are tremendously agile creatures, and they can walk on beams that are very, very thin. We're talking somewhere around 1/8 of an inch. Okay, And so what we're looking at here is the ability of these animals to traverse this beam and how quickly they can do so. What you can see is that the TBI animals have a severe deficit in traversing this narrow beam. And the animals that received Immunocal, they perform much better, almost as well as the sham animals in this really challenging motor task. So we saw a benefit there of Immunocal on preserving motor function. We also did another kind of similar assay. This is called a rotarod. And basically what this is a wheel. Okay, and so mice love to run on wheels. This happens to be a wheel that goes extremely fast. It's basically a little conveyor belt wheel that we program to go very fast. And so sham animals can stay on it for about 20 seconds before falling off. The untreated TBI mice could only stay on about half as long, but the animals treated with Immunocal showed a really essentially the same as the sham mice in terms of their performance on the rotarod. So again, Immunocal showed a nice protection or preservation of motor function in these animals that received a concussion. We also tested cognitive function in the mice. Now, it can be challenging to look at the cognitive ability of a mouse. We typically do this with mazes. In this case, we use what's called a Barnes maze. You can think of a Barnes maze as a circular platform with about 18 fake holes on it. However, one of those holes actually is real and has a little compartment that the mouse can escape into. It's called an escape pod that is below the platform. So what we do is we put the mouse in the middle of this platform and we shine a bright light on them. Mice don't like to have light shined on them. And then the mouse will try to find that escape hole. And we train them in what's called an acquisition phase over a number of days using spatial cues, basically an arrow that is drawn on the wall pointing to the escape pod. And over time, these mice learn where the escape pod is. What you can see, though, after 6 days of this training paradigm, the sham animals take about 40 seconds to find the escape pod, but the animals that received a concussion take nearly twice as long. Okay, so this is actually a measure of what we call spatial learning and memory. The animals that receive the concussion do not learn as well. But what's interesting is animals that were treated with Immunocal and then subjected to concussion, they learned just about as well as the sham mice. And so this was a nice protection, if you will, of cognitive function. We also did what's called a probe phase. This is a sort of an acute, after a week of training, we just put the mice in there and we do a certain test to see how quickly they can go to the escape area, but not necessarily enter the escape pod. And you can see that the TBI animals, the ones that had concussions, they take about twice as long as the sham mice to find that escape pod area, whereas the Immunocal treated animals, they actually were a little bit better than the sham mice at finding the escape pod. So Immunocal pre-treatment prior to concussion actually preserved cognitive function and prevented these cognitive deficiencies in the mice due to concussion. We also looked at glutathione in the cortex. So this is in the cerebral cortex, the brain of these animals. And similar to what we found in the spinal cord in ALS mice, we see with concussion a reduction in the reduced glutathione to oxidized glutathione ratio, and that reduction is prevented by Immunocal administration. So that's consistent with the mechanism that we see in vitro for neuroprotection as well as what we saw in ALS. We also looked in the brain at an index of oxidative stress. This MDA is a lipid peroxidation product that is markedly increased in the brain of animals that receive a concussion, and the levels of this lipid hydroperoxide are significantly decreased by Immunocal treatment, indicating that the Immunocal is protecting the brain from oxidative stress. Another important thing that we looked at were levels of a certain neurotrophic factor. This is a growth factor for nerve cells. It's called brain-derived neurotrophic factor, or BDNF. And it's an important factor because it protects neurons from different types of damage. And it's considered a pro-survival factor. And what we found was that mice subjected to concussion showed a marked reduction in both the pro or inactive as well as the active forms of BDNF, and that this was protected or prevented, this reduction was prevented by Immunocal treatment. So Immunocal not only reduced oxidative stress, it also protected this key pro-survival growth factor in the brains of these mice that had concussions. We also looked at several indices of what's called neuroinflammation. So in this case, inflammatory cells in the brain like microglia and astrocytes, once there's an injury, they enter that site of injury and they can cause additional oxidative stress and inflammatory stress. So in this top panel, what you're looking at here is the expression of a protein known as IBA1, which is a marker of microglia, which are an inflammatory cell in the brain. What you can see is that animals that were subjected to concussion have much higher levels of IBA1, so they have much higher microglia, and this level of IBA1 is diminished dramatically by Immunocal. And so this would argue that Immunocal actually has an anti-neuroinflammatory property in addition to having antioxidant and neuroprotective effects. I just want to make sure you guys can hear me still.
We can hear you, doctor.
Good, good, okay.
Taking notes.
Okay, good. Okay, the last two slides I want to show you, these are more looking at histology and looking at the damage in the brain that occurs in response to a concussion. This first image I'm going to show you, this is what's called the corpus callosum, which is a region in the brain where many of the white matter axonal tracts of the neurons cross. So the brain is made up of gray matter, which are the cell bodies of the neurons, and the processes, particularly the axons, which are myelinated, make up the white matter or the white matter tracts. And these axons, myelinated axons, are very important because that's how nerve impulses are conducted from cell to cell in the brain. We can stain these white matter tracts with something called Luxol Fast Blue. And so in a healthy sham mouse, you can see a nice large distribution of these white matter tracts. Interestingly, in animals that receive this moderate concussion, several days, this is about 2 weeks after concussion, you can see many of the mice lose a fair amount of these white matter tracts. And this is due to degeneration of the axons or the processes of the neurons, which of course will cut down on conduction of nerve impulses in the brain. What you can see with Immunocal is that largely these reductions in white matter tracts were prevented by Immunocal administration, and that's shown quantitatively here. So Immunocal pretreatment actually protected the neuronal axons or processes from damage. The last slide I want to show you is we looked directly at degenerating neurons or nerve cells in the brain of these mice beneath the impact site of the concussion. And we did that using a dye called fluorojade, this green dye, that only stains neurons that are degenerating or dying. And what we found was that in the brains of mice subjected to concussion, we would see foci, these areas, discrete areas where neurons were degenerating. And so we compared the fluorojade positive foci in untreated TBI animals to animals that were treated with Immunocal. And you can see that Immunocal really significantly protected these neurons from degeneration in response to a concussion. And so this series of studies really demonstrated that pretreatment with Immunocal has a really nice protective effect, increasing the resilience of the brain against a moderate traumatic brain injury. And I could mention that we're in the process of completing a study right now where we're doing a similar type of scenario, except we're using a model of repetitive mild traumatic brain injury. So this is more like somebody who maybe gets a series of concussions, maybe an athlete that over the course of a couple seasons gets 3 or 4 concussions. So it's a little bit of a different model, but I can tell you we're seeing significant benefits of the Immunocal administration in that model as well. The last study that I want to talk about is actually not published yet. We're in the process of writing this up and submitting it for publication. And this is looking in a mouse model of Alzheimer's disease. And so this again is a transgenic mouse that has expresses a human mutant gene. It's called the amyloid precursor protein gene. And this is the Swedish Indiana mutation of this gene. The amyloid precursor protein is responsible for generating the amyloid beta peptide that ultimately forms amyloid plaques or senile plaques in Alzheimer's disease. And this particular form, the Swedish Indiana mutation, leads to a high turnover rate of this amyloid precursor protein into this toxic amyloid beta peptide. So these mice, they actually develop amyloid plaque pathology just like humans that develop Alzheimer's disease. And typically it takes somewhere between 8 to 12 months for these animals to develop this Alzheimer's-like pathology. So what we did is we began giving these animals, these mice, Immunocal in their drinking water when they were 3 months old. And we gave that to them until they were 12 months old. And at that point, we analyzed their brains for biochemical, histological changes that are similar to what you would see in Alzheimer's disease in a person. And so let me start with the measure of glutathione in the brain of these animals, because that's really what we're talking about mechanistically here. And so I can just show you the ratio again. This is the ratio of reduced glutathione to oxidized glutathione in the cerebral cortex of these mice. So the non-carrier mice, these are non-transgenic control mice. These are healthy mice. These hemi-untreated mice, these are the transgenic Alzheimer's mice, untreated. And then these are the transgenic Alzheimer's mice that were treated with Immunocal. And similar to what we saw in the spinal cord in ALS or in the brain following TBI, in these Alzheimer's mice, you see a dramatic reduction in the reduced to oxidized glutathione ratio in the brains of Alzheimer's mice. But Immunocal treatment over that 9-month period of time essentially prevented that and dramatically preserved the glutathione in the brains of these animals. Now, John had mentioned a patent, something that we were looking at with this protein reelin. Reelin, let me explain what that is. It's a protein that's involved in development of the brain. However, once the brain develops, it plays a key role particularly in a region of the brain known as the entorhinal cortex and the hippocampus, in inducing something called long-term potentiation and synaptic plasticity. These are processes that underlie learning and memory. And so reelin is released by cells in the entorhinal cortex into the hippocampus, where it signals these cells to facilitate learning and memory. Interestingly, one of the first things that happens in Alzheimer's disease is that reelin expression in the entorhinal cortex goes way down, it decreases. And this likely contributes to the loss of cognitive function, the decreased memory formation that we see in Alzheimer's patients. And so if we could preserve reelin expression, that might go a long ways towards a new therapeutic approach for something like dementia. And so in this first panel here, what I want to show you is we're actually doing a process that's called in situ mRNA hybridization. Here we're actually looking at expression of the messenger RNA transcript for reelin, which will tell us which cells are actually making the reelin protein. Later on, I'll show you staining for the reelin protein, but there reelin is kind of everywhere because it's actually a secreted protein, and so it's all over the place. But in these, it's much more discrete expression because these are the cells that are actually making the reelin. What you can see in the healthy mice, so the blue here are the nuclei again, or the chromatin, just showing you all the cells in this region. And the green is stained for the reelin transcript. What you can see is that in the healthy mice, in the entorhinal cortex, there are a lot of cells that express the reelin transcript. However, in the 12-month-old Alzheimer's mice, you can see that essentially the reelin has been depleted from this region of the brain, which is what we see in human Alzheimer's patients. However, those animals that were treated with Immunocal show some preservation of reelin expression in the entorhinal cortex, and that's shown here in this graph. And so Immunocal is able to actually protect reelin expression in the entorhinal cortex. We also looked at this by a technique called Western blotting, where we're looking at the actual reelin protein. So that's in this top panel. So you can see reelin is expressed in the hippocampus entorhinal cortex of the control mice. It goes down in the Alzheimer's mice, and Immunocal again protects the expression of that reelin. We also stained the brains of these mice to look at reelin protein expression. And so here the green stain is reelin. The left panels are the control mice, the middle panels are the Alzheimer's untreated mice, and the right panels are the Alzheimer's mice treated with Immunocal. And here we're looking at entorhinal cortex, and here dentate gyrus, which is the region of the hippocampal structure that the entorhinal cortex reelin expressing neurons deposit their reelin into. And what you can see is that the Alzheimer's mice show marked reductions in reelin expression in comparison to the control mice, but Immunocal treatment really preserves this reelin expression. So you can see that here quantitatively, that these reductions in reelin are actually prevented, largely prevented by Immunocal treatment. So not only does Immunocal preserve glutathione in the brains of these Alzheimer's mice, but it also preserves reelin expression. The last thing I want to show you, because these animals have this mutation in the amyloid precursor protein and make a tremendous amount of the amyloid beta peptide, they actually form amyloid plaques. And we can look at those amyloid plaques using a compound known as amyloglow that sticks to the plaques and makes them light up. And so this green staining in the different regions of the hippocampus, the dentate gyrus, as well as the entorhinal cortex, these are amyloid plaques that have formed in the brains of these 12-month-old Alzheimer's mice. And you can see in the untreated mice, there are a large number of these plaques. In comparison, here's an Immunocal treated mouse. There still are a few plaques, but they are much smaller and take up much less area in the regions of the hippocampus. And we quantified this both by the plaque number, so the absolute number of amyloid plaques, as well as the plaque load, which is basically the percentage of the area of the hippocampus that is taken up by amyloid plaques. And what you can see essentially in all cases is that Immunocal treatment resulted in somewhere between a 50 and 80% reduction in the amyloid plaque load in these animals, which is really a tremendous effect in this model of Alzheimer's disease. So the last thing that I want to mention is that in addition to these studies that we have published and that we're working on publishing now, we've also published a number of review articles and editorials. This one was on using Immunocal to preserve glutathione as a novel neuroprotective strategy for the degenerative disorders of the nervous system. We also published this editorial or perspective article on glutathione precursors like Immunocal shielding the brain from neurotrauma. And we also had this article that we published on detailing really the effects of Immunocal in this mouse model of traumatic brain injury. And I think really to kind of summarize what we've shown is that Immunocal, through its ability to provide a precursor for glutathione synthesis, its effects in the brain and spinal cord are to increase resilience of these central nervous system areas to stress and disease mechanisms. So by preserving that glutathione, we can reduce oxidative damage, we can diminish neuroinflammation, we can preserve neurotrophic factors like brain-derived neurotrophic factor. We can also preserve key cognitive proteins like reelin. And so this combination of effects that Immunocal has really leads to really a nice potential therapeutic option, as a nutritional supplement for people. So I think I'll stop there, and if anybody has any questions, be happy to answer.
One question, Dan, for everybody. First of all, it's fantastic. I just want to tell everybody this has been 10 years worth of work, and Dr. Lindsman is the ideal partner because he's completely reliable and completely trustworthy, and every time I phoned him, he was always there to provide us the information, and it was always dependable. But then just in the context of these terms we use, if we talk about resiliency and we talk about unhealthy brain aging and healthy brain aging, how would you talk about the term resiliency in terms of unhealthy brain aging versus healthy brain aging, in discussion with Immunocal and what you've seen Immunocal do?
Yeah, so I think that gets right at the heart of one of the main theories for aging, and certainly brain aging, is it's known as the oxidative stress theory of aging, where as we age, everyone has some, decrease in our endogenous antioxidant defenses. That happens over time as we age. In addition, the mitochondria, which I mentioned are the energy producing systems in cells, they're somewhat inherently inefficient, and one of the products of mitochondrial function is a little bit of a leakage of electrons to oxygen to create free radicals. When we're young, our mitochondria are much more efficient, and we have good antioxidant stores, and so we can really limit that free radical damage. But as we get older, our mitochondria become much less efficient. They leak more electrons. We get a higher free radical load. And in addition, we have a depletion of our antioxidant reserves. And so you've got kind of two things going on there. You've got increased oxidative burden, but decreased antioxidant reserve. And this, of course, is most pronounced in the brain, because the brain, because of the way that neurons communicate and their dependence on high energy levels for neuronal communication, the brain is tremendously dependent on energy stores and is also ultra sensitive to oxidative damage. And so I think having a supplement that boosts that endogenous antioxidant pool by, in this case, providing a glutathione precursor, because glutathione pools are actually depleted as we age. That's a key way to counteract this sort of aging effect. Now, you mentioned unhealthy aging. I mean, that's just normal aging, but unhealthy aging comes in the context of other diseases that underlie the aging process. So that can be due to your particular genetic makeup, can also be due to environmental stressors. We mentioned yesterday emotional stressors, but also trauma. So if you have a history of concussion, that can lead to enhanced oxidative damage. And of course, the oxidative damage feeds into neuroinflammation, because oxidative stress triggers increased inflammatory responses in the brain, process we call neuroinflammation, and then that feeds forward in a cycle because neuroinflammatory cells produce free radicals as part of the mechanism of inflammation. And so you have sort of a vicious cycle there. So I think, having on board a supplement that can preserve and stabilize your antioxidant pools would be key for healthy brain aging.
Great, and I'll close it there and just say thank you on behalf of all of us. It was great to listen to, and I'll hand it back to John.
Ask Dr. Lindsman to rejoin us for a second. Keith, can you unmute Doctor for a second? Dr. Lindsman, would you agree with that? Is there a further definition in neuroprotective that you'd like to add for the benefit of our sales force?
Well, I think neuroprotective, I think antioxidant, anti-inflammatory, all those come to mind when I think of Immunocal. And so, neuroprotection is, I don't know if it's a common public term, to be honest with you. But I think that the basically what you're saying there is that through the, increasing the antioxidant pools, you're able to increase the resilience of these nerve cells, right, against stresses and different types of damage. And so that's the kind of the underlying definition of neuroprotection.
And my other question on there, Doc, for the benefit of not only myself, but everyone else listening, I remember at least 10 years ago, might be even 20 years ago, Time Magazine had on its cover for the first time about inflammation. That inflammation was linked to comment on that if you would. Inflammation and how important it is to get the inflammation down, not only for the brain obviously, but for the rest of the body. But if you can kind of give us a Reader's Digest on that.
Yeah, I mean, and inflammation has become a very public term, right? You can't turn on the TV without seeing something about decreasing inflammation, whether it be using something to decrease brown spots on your skin. That's a type of inflammation and oxidative stress that causes that. In terms of aging, of course, inflammation increases with aging. That's why we have so many problems with our back and our knees and our brains, right? That's largely due to oxidative stress and inflammation. And so I think the interesting thing about Immunocal is that the anti-inflammatory, or in my case, I work on specifically neuroinflammation, those effects really come from the antioxidant boosting effects. So inflammatory cells like microglia, astrocytes, or even certain types of white blood cells, one of the mechanisms by which they become inflammatory is they undergo mild oxidative stress. That's an underlying mechanism of inflammation within the inflammatory cell. And so probably what's happening is Immunocal, through its antioxidant action by boosting glutathione, is decreasing the oxidative stress within the inflammatory cells and diminishing their ability to take on that neuroinflammatory phenotype.
So that's...
But John, you know, as we're coming into these Zoom calls and we're doing all these, they're all connected. You know, today was about don't lose your mind, take Immunocal. And next on August the 8th, it's going to be take Immunocal so you're still able to walk. And then we're going even further after that, you know, so it's mind, body, soul, and all around good health, right?
Well, we have so much content and in-depth analysis with regard to how Immunocal acts. And this is the great asset of this company from 40 years ago, not only about how the product acts, but actually, you know, why we make the product the way we do. And it's all from insights that we've gained from doing our own research and collaborating with people like Dr. Lindsman and Dr. Nathan Lewis.
Well, you know, I'm so looking forward to thank you for today. It was just amazing. And where we're going on the 8th is even